All right, today we are going to be talking about Punnett squares and learning how to complete a Punnett square. A Punnett square is a diagram that's used to predict the outcomes for offspring when we cross two different organisms. Before we start talking about Punnett squares, we kind of need to keep in mind some of the vocabulary that we've been talking about in the genetics unit. The first word is trait. So a trait is a specific characteristic that varies from one individual to another. Some examples of traits would be hair color, eye color, height, curly hair, straight hair, wavy hair, whether you have a hitchhiker's thumb or not, widow's peak or not, attached earlobes or not. All of those are examples of traits. An allele, alleles are different possibilities for a given trait, so every trait has at least two alleles. So eye color, you actually have four different options. You can have brown eyes, blue eyes, green eyes, hazel eyes. Those are all represented by different letters there. Genotype versus phenotype. Genotype is the genetic makeup of an organism. So the allele combination that an organism has, and those alleles are shown by letters. So the example of a genotype would be capital T, lowercase s, capital G, capital W, lowercase w, any letter combination. Capital letters are going to be the dominant and lowercase are going to be the recessive. Phenotype, that's the physical characteristic of an organism. So the way that an organism looks. So when we look at someone, we can see they have curly hair, they are 5'6", they have blue eyes. If we're looking at plants, we can see the stem height, the flower color. Those are all things that we can see. Things like blood type are also going to be considered phenotypes, even though we can't necessarily see someone's blood type just by looking at them. But those would still be physical characteristics. Dominant and recessive, these are going to be really important when we're talking about Punnett squares. Dominant alleles are going to be shown by the capital letter. So you can see we have the capital R here to show the dominant allele. It's going to mask the other trait. So if there's one capital letter there, it doesn't matter what the second letter is. It's automatically going to show what that dominant trait is. So if that dominant trait is brown hair, if we have one capital R, then that brown hair is going to be shown. In order to show blonde hair, we would have to have two recessive alleles, and that would be the lowercase letters. So someone with blonde hair would have lowercase r, whereas someone with brown hair is going to have capital R, or capital R, lowercase r. So a dominant one, we just need one capital letter to show that dominant trait. To be recessive in something, you need to have two lowercase letters. This just kind of goes through an example. So using the letter T, so capital T is going to be a widow's peak, which is the first image there on the left, where the hair kind of comes to a point. And then lowercase t is going to represent a straight hairline, so you can see that on Reese Witherspoon there. And if an individual has a capital T, they're going to show that widow's peak because it's dominant. If they have a capital T, lowercase t, they're also going to show that widow's peak because they have one of those capital letters. And we call this complete dominance. So there will be exceptions to this rule. We'll talk about that a little bit later on. Lowercase t, that represents an offspring or a person with a straight hairline. Homozygous versus heterozygous. This also is going to be extremely important when we're talking about Punnett squares. It's important that you know the difference between homozygous and heterozygous. Homozygous is going to refer to an organism that has two identical alleles. So that can be capital or lowercase. And again, the letter doesn't matter. The example that's given here is capital T, and lowercase t, or capital R, and lowercase r. If we recall what we know about prefixes, homo means same. So it makes sense that we're talking about the same capital or lowercase. And then heterozygous, when we're talking about the prefix hetero, we know that that means different. So An organism that is heterozygous is going to have two different alleles for the same trait. So capital and a lowercase. And if you look at our example, we have capital T, lowercase t, and capital R, lowercase r. The lettering there, when we write these, we want to put our capital letter first. So it'll go capital lowercase instead of lowercase capital letter, just for some housekeeping. Punnett squares. So a Punnett square is a diagram that shows the genetic combinations that might result from a genetic cross. So if we cross this female with this male and we're looking at this one specific trait, what are the chances that their offspring are going to show that trait or not show that trait? Probability, that's the chance that something will happen. So when we look at a Punnett square, we have 4 possibilities there, and if they happen to have 4 offspring, that doesn't automatically mean that they're going to show the same percentages that we predicted they would show. So this is how we set up our Punnett square. Parent number 1 is going to go on top, and parent number 2 is going to go on the left-hand side. One of their alleles goes over each box. So you can see in this example, we have 2 parents. Each one is heterozygous for that trait, and we're using the letter A. So you have capital A, lowercase a, and capital A, lowercase a. And then the squares down below, that shows us the possibilities for our offspring. So in this case, we have in the first box here, you have a 25% chance of having an offspring that is homozygous dominant. And then if we look here and here, in the second and third square, you have a 50% chance of having an offspring that is heterozygous dominant. And then our last square here, you have a 25% chance of having an offspring that is homozygous recessive for that trait. And we don't know what that trait is right now. We're just looking at how to set up our Punnett square. So the next slide here kind of shows us how to, what I call, drag and drop those letters. So the top parent, you're just going to pull those letters down into the box that's directly below them. The side parent, you're going to pull them to the right, drag and drop. And that leaves you with 2 alleles in each square there. This example, so we're looking at yellow and white. That's our trait. So I'm assuming that would be a flower color. So you can see each parent is heterozygous for this trait. So each parent has a 50% chance of passing on a capital Y, and each parent has a 50% chance of passing on a lowercase y. So when we put our parents into the Punnett square, it doesn't matter which parent goes on top and which parent goes to the side. But you can see our, once they've been dragged and dropped, our genotype is, the possibilities that we have would be capital Y, capital Y, capital Y, lowercase y, and then lowercase y, lowercase y. And our phenotype, these 3, any of them with 1 capital letter, are going to show the yellow trait. And the only one that's going to show white would be our homozygous recessive, this last square here, and that would be a 25% chance of a white trait showing. All right, so let's go ahead and do another example. In this one, we're going to use the genotype for the parents as capital T, and lowercase t, lowercase t. We're talking about plant stem height here, so T equals tall, or capital T equals tall, and lowercase t is going to equal short. So for the parents here, our first parent there is going to be homozygous dominant for tall stem height, and our second parent is actually going to be homozygous recessive, so short stem height there. So when we put them in our Punnett square, you can see when we cross it, we have parent number 1 on top, so capital T, parent number 2 to the left, lowercase t, drag and drop those letters there, and we end up with actually all of our genotypes being capital T, lowercase t. and we would have a phenotype that would be 100% tall. So the probability that our offspring are showing that tall stem height is 100%, and they would all have the same genotype. Keep in mind that each box is worth 25% because it's going to add up to 100% total. Hopefully this gives you a better understanding of how to complete a Punnett Sqaure so that when you're completing your homework today, you have a better grasp of